Thank you very much to the organizers for giving me the opportunity to speak today. My name is Lauren Sanders. I'm a staff scientist at NASA GeneLab, and I also help lead the Artificial Intelligence Space Biology group. Today I'm going to be talking about machine learning for space biological imagery data. I wanted to start out by showing again the image shown here by my colleague Ryan Scott in his previous talk. This shows the open science data repositories at NASA, including GeneLab and the Ames Life Sciences Data Archive. In the AI/ML group, we emphasize reusing and reanalyzing space biological data from the open science data repositories for knowledge gain, prediction, hypothesis generation, and ultimately countermeasure repurposing for safer spaceflight. I'm going to be talking about two efforts. One, to generate benchmark training data sets with space biological imagery data, and another effort using deep learning to analyze microscopy data. There are multiple challenges and opportunities for AI analysis in space biology. AI has been used to great effect for analyzing large biomedical data sets, but space biology has its own unique constraints. These experiments sent to space tend to be very expensive, and so they end up being small with low sample number. There's also an emphasis on the transfer from model organism to human. Space biological data sets tend to be heterogeneous. We have data sets that have omics data and images and environmental variables all in the same experiment. That brings me to the need to generate benchmark data sets so that we have AI-ready space biology imagery data to benchmark and train new models. This will allow us to measure the performance of new models or models that we repurpose from other domains, and we already have an AI-ready data set ready to go for these trainings. This will also allow us to standardize AI readiness principles for each type of space biology imagery data so that all data sets are formatted and labeled the same way. This will allow us to develop new models specifically for space biology challenges and then evaluate standard metrics of model performance on the same data sets every time. The NASA Science Mission Directorate has called for the creation of benchmark data sets from each division. And so our division, the Biological and Physical Sciences Division, is delivering 3 data sets this year. One is an omics data set, and then we have 2 image data sets. I'm going to be focusing today on the microscopy data set that we are generating, which is a radiation exposure microscopy data set with DNA damaged events stained as distinct foci. And this data set comes from Dr. Sivan Costas from the Radiation Biophysics Lab at NASA Ames. The scientific motivation for this data set is that exposure to radiation causes DNA damage to cells. And we know that spaceflight exposes cells to increased radiation and DNA damage risk. We can analyze this risk by looking at radiation-induced foci, which show up as discrete markers of DNA damage in a nucleus. We can study deep space radiation on Earth without needing to send samples to space, and in this way we can generate larger data sets. So we know that space radiation is composed of solar particle events and galactic cosmic rays mainly. And solar particle events are rare and difficult to predict, so we focus our efforts on trying to understand the effect of galactic cosmic rays, or GCRs, on biological systems. GCRs are composed of several different components, and in particular we're going to be focusing on these high mass charge particles since they cause the most damage to DNA. We simulate the effect of GCR particles on living systems by performing biological experiments at the NASA Space Radiation Laboratory in Brookhaven National Lab, where there is a particle accelerator for GCR simulation. So particles are accelerated in this ring accelerator you can see here on the top until they reach the energy we're interested in, and then they're siphoned off and sent off to the biological experiments. So that's how this data set that we're using for the benchmark data set was generated. This data set was motivated by the scientific question, how are human immune cells affected by simulated space radiation? So blood immune cells were taken from 761 human volunteers and subjected to 3 different particles with increasing energy, and then also subjected to gamma rays as a control. Microscope images were taken at baseline or 4 or 24 hours post-radiation and stained for a marker of DNA repair. Now that we have this benchmark data set, we want to employ AI/ML approaches to analyze the underlying patterns of DNA damage and repair. So our experimental design for this project was we wanted to focus on the highest energy particle, iron, first, and compare it to the gamma control, and understand the difference between these two conditions in the radiation-induced DNA damage. Our input were these light microscopy images, which had been cropped to include 1 nucleus per image and were stained with the 53BP1 antibody to identify DNA repair sites. And right now we're focusing on the 4-hour time point. We designed a machine learning pipeline with 2 sequential classifiers. One was a QC classifier, so that we could exclude any out of focus or abnormally imaged nuclei. So we labeled the QC pass and QC fail, and then trained the classifier on those. Then the clean outputs of the QC model were fed into a radiation classifier to distinguish between iron and gamma based on the DNA damage pattern. The model that we're using for this analysis is a deep learning model called the Self-Supervised Learner, developed by the SpaceML group. And this is an interesting application because we're using a cross-disciplinary implementation of this deep learning model. SpaceML specifically develops deep learning tools for astrophysics and space science applications, and here we're repurposing their SSL tool for a space biology application. SSL is special because it enables self-supervised pre-training with only a few labeled examples. There are 2 steps to training SSL on image data. First, there's a self-supervised pre-training step where no images are labeled and you just feed in a very large unlabeled data set. This allows the self-supervised model to learn features characterizing just what do microscopy images look like in general. Then we run a classifier which we fine-tune to predict our desired label. So we have a few labeled examples of QC pass or fail or iron or gamma. Now the way that this works is through contrastive self-supervised pre-training, which is a training method that trains a model on augmentations of the same image. So the model sort of trains itself to maximize similarity between pairs of augmentations from the same image. In order to perform this analysis, we first had to make this microscopy data set AI ready. Each microscope image started off as a Z-stack with either 9 or 21 indices, and each image was 32-bit, whereas a 16-bit or lower is nice for most machine learning applications. So we generated an AI readiness pipeline where images were converted to a 16-bit maximum intensity projection using the diplib library in Python. So the first thing that we wanted to do with this analysis was to understand whether we wanted to use a pre-trained model or not. And so we wanted to see whether we wanted to use something called transfer learning, which is a machine learning method where a model developed for a task can be reused as the starting point for a model on a similar task. This can be a good thing because it can save resources and improve efficiency. So we had access to ResNet18, which is a well-known 18-layer convolutional neural network, not pre-trained. But ImageNet ResNet18 is the ResNet18 neural network model pre-trained on more than 1 million images from the ImageNet database. So it already kind of knows what a picture looks like in general, not a microscope picture in particular, but just images in general. So when we trained our classifier using either the ResNet18 or the pre-trained ImageNet, what we found was that our QC classifier did perform better with the pre-trained ImageNet, whereas our radiation classifier, both of them performed about the same. So for future steps here, we'd like to understand why the QC classifier worked better on the pre-trained model. So the biological conclusions from the study so far, when we took a look at the number of QC pass and QC fail images from gamma and iron, what we noticed was there was a much higher percentage of fail images in for the iron data set. So what this tells us is that these higher energy particles result in much greater overall cell damage, because when you hit a cell with these higher energy particles, they tend to be so deformed that they actually lift up off the plate, which ends up with more blurred images. So we can already tell that the higher energy particles result in much greater DNA damage. The next thing that we want to do is go back to the data set and take the argon and the silicon particle exposure data, which are higher energy than gamma but lower energy than iron, and we want to see whether they're more similar to iron or whether they're more similar to gamma. So as particles, we might think they'd be more similar to iron, but as low energy, they might be more similar to gamma. So this will inform future experimental design, experimental interpretation, and then decision making. How do we think about galactic cosmic rays, where most particles might be lower energy? We also have several next steps we want to investigate on the AI side. We want to perform more hyperparameter tuning and see whether we can get better results. We want to employ explainable AI techniques where we'll be able to identify what are the regions or the aspects of the image that are informing these classifications. So we can maybe identify whether it's some kind of cell deformity or the DNA damage track patterns. We also want to develop an image similarity search using SSL so that we can accelerate our ability to test hypotheses and identify similar images. And then identify a web platform so that users can find similar images. I finally would like to acknowledge everyone in our group in the Radiation Biophysics Lab, the AIML Science Mission Directorate Working Group, the SpaceML Group, and the Frontier Development Lab, our compute resources, and of course all of our funding support. Thank you very much.